Hello, good morning everybody. So, today we will still continue the most significant role of vanadium in biology. So, we will try to find out some of the biological roles that vanadium can play in different organisms, because it is not very much available in human system, but it's certainly beneficial to some of these different organisms. And these organisms can see whether it can be useful for some chemical transformations also. That's why today we will be just talking about this second part of the system, which is the vanadium enzymes. And this vanadium enzymes second part will deal with some of the things what we have already discussed, that the vanadium haloperoxidases which catalyze the oxidation of halides. So, if we have some vanadium bearing chlorine peroxidase or vanadium bearing bromine peroxidase, so definitely they all will have the corresponding oxidase activity, which is very important. So, how nature is performing this dual role of activating some of the substrates in presence of the halides, which are very much available in marine system or in sea water. So, we all know that in sea water large amount of the halides, whether they are bromide, chloride or iodides, are available. So, if we have any such thing, that means whether you can have bromide or chloride or iodide, and how we can show some oxidase activity or peroxidase activity rather. So, this is basically not the typical oxidase activity, these are the corresponding peroxidase activity. So, if the thing is reacting with O2 minus, so what is happening therefore? We know that these peroxides can function in both ways. That means it has intermediate oxidation state compared to molecular oxygen and the superoxide when we can have. So, this basically when go for disproportionate reaction, it can go for oxidation as well as reduction reaction. But since we are talking about the peroxidase activity, so we will be looking for the corresponding oxidation reaction on the halides. So, if we are able to oxidize the chloride, the bromide or the iodide, we will be able to see some peroxidase activity when peroxidases are present in the system. So, these basically use the hydrogen peroxide, and vanadium haloperoxidases are therefore classified according to the most electronegative halogen oxidized. So, if they are oxidizing chloride, we call it as a chloroperoxidase. If it is oxidizing bromo, it is bromoperoxidase, like that. So, depending upon the electronegative halogen, that means the halide anion, they have the different roles. and we all know that the redox potential for the corresponding oxidation of iodide, bromide and chloride are different. So, we need different E0 values, the redox potential values for the system, which is again getting activated only by the presence of peroxide anion. So, how these peroxide anion can be modulated for different types of oxidation reactions on chloride, on bromide and iodide, that we will see, which is intervened by the presence of vanadium center. So, one such example is that our vanadium bromoperoxidase, which can be isolated from marine algae including chlorophyta, which we all know as the green algae, and phaeophyta, which is brown algae, and rhodophyta, the green algae. So, the origin is basically the algal origin, and this algae origin is therefore useful if we want to see something, that means the reactivity related to the bromide anion. So, if certain reactivity we can talk in terms of the typical organic chemistry also, that whether we can go for some reaction where we just simply see, that means we call in the model system, that we will see afterwards, that if there is some model system where the system can show some bromoperoxidase activity, that means we just simply supply or provide bromide anion and the hydrogen peroxide and some vanadium based catalyst. So, if that vanadium based catalyst can show some reaction on these and we able to generate some bromonium ion, and that bromonium ion can attack the ring and can end up with some generation of some bromobenzene derivative. So, this is also helpful, that whether we should use some vanadium based catalyst for this sort of transformations, because large number of reactions are already known in chemical literature for the organic transformation, that means the bromination of the substrate, any kind of bromination, chlorination and iodination reaction. But when we find out something where the biological system is also helpful for giving some of these derivatives, the brominated products which we get from seawater, these brominated products, how the nature is doing for us, all these algal bromoperoxidase or chloroperoxidases are doing that thing. So, that can be also utilized if we can have a corresponding vanadium based catalyst in our hand. So, all these brown algae, red algae and green algae can give rise to some compounds where we see that the overall reaction what we can have for vanadium haloperoxidases, which basically catalyze the reaction of RH transforming to RX. That means, if we have benzene, the benzene will be converted to bromobenzene. With the reaction of bromide anion, X minus will be there, that means Br minus will be there, hydrogen peroxide will be there, and some proton source should also be there. So, if it is present from the enzyme, that means some pocket is there where the vanadium is nicely trapped inside. And last time what we have seen in our last class, that these pockets basically are very much well fitting with the corresponding vanadate anion. So, the vanadate anion and the corresponding surroundings can give rise to some of these, that means the terminal ends of the enzymes of the protein chain can also provide some of these protons for this complete transformation, because this X is incorporated nicely within the molecule RH, converting it to RX, and one molecule of hydrogen peroxide with two other hydrogen atoms produce 2 molecules of water. So, how we get that particular reaction, we will just see for a typical transformation where we can have Br minus is there, which is reacting with H2O2, and our catalyst is vanadium bromoperoxidase. What is forming over there is Br plus, which is written within the inverted commas like that, is like Br plus like intermediate, because it is not that only Br plus is formed, but some of other species like HOBr, where we can consider formally that HO is the minus part, the negative part, and Br is the positive part, then is Br plus. So, these and other type of intermediates are forming along with generation of some amount of bromine, because we all know that when bromide ion is getting oxidized, we can produce in situ, that means within the reaction medium we can produce some of these free elemental bromine. Like that, when we react Br minus with potassium bromate, that also liberates bromine. And when this basically, so synthetically in the laboratory, when Br minus is reacting with some oxidizing agent like bromate anion, say potassium bromate. So, when it is reacting with potassium bromate, it basically liberates bromine. So, whether we can have the bromonium ion, what is written in the reaction, or we have the free elemental bromine, which is utilized for the typical oxidation of the substrate. So, you have the substrate and that substrate is getting oxidized, and with the incorporation of bromine within the ring. So, once this is a very straight cut and very simple reaction we find in the laboratory, when bromide is reacting with potassium bromate, bromine is getting eliminated, and if these two, when we have Br minus and Br plus, so that immediately gives us Br 3 minus also, and some organic part which is also getting brominated. So, basically what we should focus our attention, that whether we can have this Br plus in the system and how this can be identified. So, any characterization pattern for that, is we need some amount of characterization data for the identification of Br plus, and this particular species is only responsible for the transformation or the reactivity or the catalytic reaction related to vanadium bromoperoxidase. So, when along with all these, like this Br plus as the intermediate or the other species is forming, we can have two different steps of reactions when the organic substrate, this is the substrate like that of our simple benzene ring, when benzene ring is reacting, whether we will be getting some bromobenzene or not. So, some suitable organic substrates like that, what we have seen in our previous class, that all brominated marine chemicals, the marine natural products we have, they are mostly and very useful because they have the potential for use as good pharmaceutical chemical and good other molecule. So, which has the corresponding brominated organic compound. So, if we have the substrate, so any useful substrate, any medicinally important organic substrate which can be more useful if we can go for a typical bromination reaction. So, this substrate is getting brominated when the substrate is suitably available and it has certain sites where the bromination can take place. That means, it has some useful CH bonds available, where CH bond can be substituted by CBr bond. So, when substrate is present, we get definitely we will get the corresponding brominated derivative of the substrate. And in this particular case, when this substrate is not present and we have more amount of hydrogen peroxide as well as water, we can generate singlet oxygen from the reaction system. So, basically this is also a conversion. If we want to produce some amount of singlet oxygen, what we all know that the molecular oxygen in the ground state, what is available in nature in air, they are all triplet oxygen. But for some transformations we want to convert the triplet oxygen to the singlet one. So, this reaction also goes for the conversion of this triplet oxygen to singlet oxygen, and Br is generated as Br minus again. So, this is the simple and straight cut reaction mechanism for the conversion of this particular reaction for the bromination, and we get this particular information from that Coordination Chemistry Review reference of 2003. So, now we will be interested to know that what type of environment, because last time what we have seen that we just shown you that we can have a typical pocket, and this pocket was there and this pocket was surrounded by so many amino acids. So, in that particular case what we have seen that if we can have initially in the enzyme backbone one pocket of vanadate anion can be formed, which nicely trap the tetrahedral vanadate anion inside. So, we can trap VO4 3 minus, vanadium is present in +5 oxidation state, and these oxygens, that means the vanadium oxygen. So, whether they will be further reacting with some proton or some other substrate, that will be dependent on how they are encapsulated by the different amino acid. So, if we see that there are several amino acid residues which can surround this particular pocket. So, sometime we will find that 1, 2, 3, 4, 5, 6, 7 amino acid residues with terminal ends are sufficient to encapsulate the vanadate anion within, and this particular one definitely will show some interaction with the amino acid residues. So, what are those interactions? That we will now see that if we have this vanadium oxygen bond and oxygen is rightly nucleophilic in character and it has some delta negative charges. So, it will start interacting with the amino acid residues such that through oxygen it is basically getting stabilized within this particular pocket. So, in the native state what we will find that you have this particular vanadate anion was there, and this vanadate anion was in tetrahedral geometry, and this tetrahedral geometry can go for some interesting conversion of structure. That means, it can immediately go for a trigonal bipyramidal geometry. This particular geometry around this vanadium is trigonal bipyramid. So, this is the trigonal plane, this O3, the 3 O atoms in this particular basal plane is a trigon, and these are the two apical sides. When one apical side is occupied by the OH and another one is been occupied by the protein chain through the nitrogen of histidine 496. So, this basically the coordination of this particular nitrogen from the histidine side chain, which is very important and is well known and famous for that also, that whenever we have a protein chain and that protein chain can go for the corresponding coordination to the vanadium site. And this particular one, what we see also in case of hemoglobin molecule, that in case of hemoglobin molecule we have this particular porphyrin binding to the iron site, and the fifth coordination site was occupied by the nitrogen from the globin chain. The same thing is happening, that the histidine nitrogen, the imidazole nitrogen from the histidine residue of the protein chain is very much useful for showing some strong interaction with this vanadium. So, this vanadium nitrogen bond is established, and that basically controls the reactivity pattern of the entire system. So, when it is forming an apical bond to the vanadium side, so this is the new bond which is forming apart from the 4 existing vanadium oxygen bonds. So, in the vanadate anion what we had 4 vanadium oxygen bonds present there, and now the new bond is forming. Since the conversion is from a tetrahedral to trigonal bipyramidal, we do not have any other option, that means square pyramidal geometry is also not forming. And interestingly, when we have this kind of arrangement, we expect that this vanadium nitrogen bond can be little bit longer compared to the other vanadium oxygen bonds. So, this particular coordination is basically very important and is a vital coordination for converting the vanadate anion to a trigonal bipyramidal system or center based on vanadium, where it is establishing or showing a corresponding bond with that of the nitrogen from the histidine residue. So, this was basically the other end of the apical bond, which was our OH, and this has been converted through some proton delivery on the vanadium oxygen bond of the vanadate anion. So, it can come from the corresponding residues which is available from the protein chain, and then it can show, since OH is there and since this histidine 404 which is quite far from histidine 496, that this histidine residue is not coming close in contact with this particular side, such that it can form some other type of bond with that of the nitrogen, replacing this OH as water molecule. So, it can go for stabilization of this particular unit. So, VOH part, this VOH part is getting stabilized through hydrogen bonding interactions with the histidine residues. So, as we all know that in histidine we have that imidazole side chain, and this imidazole side chain basically go for one as tertiary nitrogen and one as NH. So, this particular nitrogen with a lone pair of electron, most of the time it can bind our iron site. Here also the way it is forming the bond, the vanadium nitrogen bond is forming by the coordinate bond coming from this tertiary nitrogen. So, this orientation of this histidine residue, so is this histidine residue, whether it is 404 or 496, the number is 404 or 496. When it is 496, it is showing this particular bonding arrangement. When it is 404, it is useful for stabilizing the VO bond. That means, if we have the VOH bond if the available nitrogen is the same one, so it can show hydrogen bonding interaction with this hydrogen, or this can use this entire NH function such that it can go for some interaction involving this hydrogen. So, we can have two different options, whether this nitrogen is available which is not engaged in any other kind of interactions such that bonding with the vanadium, it can show hydrogen bonding interaction taking the hydrogen of the OH, or this NH can show the same hydrogen bonding interaction with this oxygen. So, this histidine can have dual roles to play. It can function as a hydrogen donor, hydrogen bonding donor, basically hydrogen bonding donor. It can function as hydrogen bond donor, as well as it can function as hydrogen bond acceptor. So, whenever we have any histidine residues in the protein chain or the polypeptide chain, we know it is very useful to stabilize the vanadium center within the pocket. So, these two apical bonds are therefore stabilized, one through direct coordinate bond and other through hydrogen bonding interactions. And in this particular case, this OO, so these two oxygens on the right hand side would be stabilized by showing some bifurcated hydrogen bonding interactions, the NHO and NHO bifurcated hydrogen bonding interactions we call, because simultaneously this oxygen is functioning to two hydrogen bond donors, which is coming from this arginase 360 and arginase 490, and this NH can show either interaction with this, and other end also can show interaction with this. So, basically this is a rigid backbone on arginase unit like urea or bigonide. This is the bigonide end. So, this bigonide end basically functioning as a bridge like that of bridging two ends. We all know that some of the bridging groups are available all the time, like the simple copper acetate binding. When we have acetate groups CH3COO minus, so this acetate group can be a very useful bridging unit to a binuclear system. If it is present in copper acetate, it can show the bridging interaction between these two copper centers. So, this particular entity is a very stable entity. So, this particular motif is getting stabilized through bridge. So, we can call it as a stabilization through bridge. So, this stabilization through bridging interaction is also seen in case of our this vanadium. So, vanadium as these two oxygens is already attached to vanadium. So, this dioxo vanadium unit in cis position, how we can stabilize this? So, this can stabilize with this NH unit of arginase 490. So, if we have a rigid backbone on arginate unit, which is very much similar to that of bridging unit of the acetate group, then this particular entity is getting stabilized through hydrogen bonding interactions with these NH functions. So, this bigonide type of part on the arginase unit is very useful in stabilizing the adjacent oxo units on vanadium. That means, the cis dioxo unit is getting stabilized through hydrogen bonding interactions. So, this part is stabilized and it can show also for some more interaction with serine residue, serine 402, and the other oxygen is showing again bifurcated interactions with NH of lysine and NH of glutamic acid. So, all these hydrogen bonding interactions, so 4 of these oxygens are stabilized through hydrogen bonding interactions. That means they try to remain on the vanadium center. Only this vanadium nitrogen interaction is unique one, is a different one, which is showing a direct vanadium nitrogen coordinate bond, and this particular bond is therefore the most stable entity which can hold this vanadium to the protein chain. So, during the reaction within the catalytic cycle, what we will see that during the reaction all these are vanadium oxygen interactions. When we have the vanadate anion, we consider the bonding between vanadium and oxygen is less than vanadium oxygen double bond, is more than vanadium oxygen single bond, the bond order, but it is less than vanadium oxygen double bond. But in this particular case, when the catalytic cycle is on, the catalytic cycle is operating and it is mostly stabilized through this end. Still you have the still histidine 496 nitrogen interaction with the vanadium, and we have arginase 360, this interaction arginase 360 is also interacting with this oxygen, but all other oxygens are not present. Instead we have a peroxo linkage. So, within the catalytic cycle we have the intermediate as the peroxo intermediate. So, vanadium is showing that peroxo intermediate through the reaction with hydrogen peroxide, and that vanadium peroxide unit, rather vanadyl peroxide unit, can be very easily identified through NMR experiments also. Vanadium NMR we can do, and that vanadium NMR can establish that both the vanadyl oxygen is present, which is stabilized by hydrogen bonding interaction, as well as the peroxo unit, which is also stabilized by the glycine amide backbone of NH and the lysine 353. So, the conversion from the native state, which is trigonal bipyramid in geometry, to a square pyramidal geometry, this geometry is completely different and is also well known and well established geometry for so many vanadium sites where we have the vanadyl as the species. So, we can have in synthetic molecules also, when we will go for studying the model compounds, we will see that these two are the corresponding bonds which are basically the cis bonds, and this is also another cis bonds, basically giving a square pyramidal geometry. So, any bidentate ligand like acetyl acetone or any other, so this can be occupied by bidentate ligand, and this can be by O2 minus, the peroxide ligand. So, we immediately get a corresponding square pyramidal geometry from the native geometry, which is trigonal bipyramid in nature, and because we can have this particular arrangement, that means this arrangement is already there when we have the trigonal bipyramidal geometry, because this is the trigonal bipyramidal geometry, it has also the two cis positions. So, if we can have these two cis positions, so we can justify that these two cis positions can be occupied by any bidentate ligand or the peroxide linkage, only the rearrangements in all other sites are taking place, because this is the new connectivity, the vanadilyl ion, such that within the species we have this interaction, the vanadyl plus peroxide interactions, which can be found out from NMR study also. So, after identifying these two centers, one is the native center and another is the reactive peroxo site, we can go for getting the entire catalytic cycle, which is very important to note that when we have, that means this is the well known thing, that means what we have, this is the thing, that means the trigonal bipyramidal geometry. This trigonal bipyramidal geometry we can start with. So, we are not talking in terms of the corresponding hydrogen bonding interactions through all these oxygen atoms. So, this is the corresponding trigonal bipyramidal geometry, and this particular geometry can show very useful interaction with that nitrogen already. So, this particular bond basically controlling the reaction pattern on the vanadium center. So, we have these hydrogen bonding interactions with this OH also. So, when it starts reacting with hydrogen peroxide, so showing the peroxidase activity. So, this particular peroxide can interact, so that means it goes for reacting this vanadium site through this particular position, because we can have more vacancy on this particular side, because this side is little bit crowded. So, this side we can have, and during this particular reaction with hydrogen peroxide, the one proton from the hydrogen peroxide unit is coming, and that proton is going to attach this particular OH function on the vanadium, making it vanadium hydroxide bond to a vanadium aquo bond. So, this OH function attached to the vanadium is also basic function. So, basicity of this OH group is such that it can abstract one proton from hydrogen peroxide to convert this vanadium hydroxide linkage to vanadium aquo linkage. So, this proton from hydrogen peroxide goes on this oxygen, and we are remaining with hydroperoxide anion. So, this hydroperoxide anion then just go and attack this particular vanadium site. So, this particular attack on the vanadium site is already protonated. So, this water molecule will go immediately from there, and this OH function will also go through another step of protonation. This hydrogen will go and attack this particular OH, and it can go for another water molecule. So, removal of the VOH bond, that means the vanadium oxide bond, vanadium oxido bond, is very useful, because the basicity of these OH functions attached to the vanadium is more, and this particular basicity can also transfer the second proton from the hydrogen peroxide to this OH group, and it is converted to water molecule. As soon as it converted to water molecule, it goes out from the vanadium coordination environment, because we have the competitive peroxide anion on our hand, and that peroxide anion can basically remove this particular bound water molecule, which is loosely bound to the vanadium center, and we expect what we see here also, that it can go for the corresponding ligand exchange reaction. So, the ligand exchange reaction for this OH by peroxide anion is taking place, and we are ending with the corresponding square pyramidal geometry. What we have just now we have discussed, that this is the vanadium square plane, where we have the already present OO bonds were there, and two new bonds from the peroxide unit is forming, and the apical site is occupied still with that of the nitrogen of the protein chain of the enzyme. So, this is the peroxide intermediate. So, for all these reactions, the basic step for the formation of this is that we should generate the corresponding peroxide intermediate to react it with the chloride anion. So, vanadium can show the mechanistically the vanadium chloroperoxidase activity can be seen when hydrogen peroxide is first reacting with the vanadium center, and this reaction with the vanadium center can give rise to very nicely the corresponding vanadium peroxo compound, which is well known to the laboratory also. In the analytical chemistry laboratory, we see that vanadium peroxo compounds are very characteristic, having very characteristic color. It shows some beautiful coloration reaction with the peroxide anion. That means vanadium always has the tendency to form vanadium peroxo bonds, whether it is forming in the enzyme state or it is forming in the test tube in the laboratory. So, in the next step, once the vanadium peroxo linkage is formed, the chloride anion from the water source, from the sea water also, that large amount of sodium chloride, potassium chloride, magnesium chloride we can have, and all these chlorides as the corresponding chloride anion is then reacting on this OO peroxide linkage on vanadium, and is basically at this particular point, because here we have the trigonal bipyramidal geometry, and that particular bipyramidal geometry has been squeezed basically to a square pyramidal geometry, and for that purpose these two coordination position has gone. Instead of that, we have in plane coordination, which is forming forcefully, because the peroxide linkage has only this alternative, because it cannot have a basal apical bonding, only it can have both the vanadium oxygen bonds in the basal position. So, this OO bond which is in the strained form, because the vanadium oxygen is a 3-membered ring which is not very stable and not very strong. So, once Cl minus can go and attack this particular peroxo linkage, the OO bond is getting cleaved, and once this OO bond is getting cleaved, what we find that this oxygen is again revert back to the original apical side, and this oxygen is taking the position of this trigonal face. So, this, if this is the driving force, but is not happening altogether, what is happening that this OO bond is getting cleaved and forming this vanadium OH bond and vanadium HOCl bond, because this H is coming and we can get the corresponding reaction with water molecule. So, these water molecules providing some extra proton, because we need so many protons as well as the hydroxide groups. So, in a single step, when vanadium peroxo compound is getting cleaved by the attack of the Cl minus in presence of proton as well as water molecule, we get this particular step or this particular intermediate followed by the peroxo intermediate, where hypochlorite that means in the form of hypochlorous acid, HOCl is the hypochlorous acid. So, hypochlorous acid is generated from the reaction of hydrogen peroxide and Cl minus, and one such bond is converted to OH and another one is coming from the water molecule as OH, because what we have in the native state is the OH bond connected to vanadium in the apical side. So, this particular part is getting regenerated. Similarly, these two oxygens we are not touching. So, they are the silent spectators in the catalytic cycle. So, we are not touching these two oxygens. So, they remain as this. Only the nitrogen is also controlling the entire reaction process. So, we can have these three sites. So, in the case of model compound development, the model studies will find that if we can have one particular ligand system where all these three positions, these facial positions, these nitrogen, oxygen, oxygens, these are the facial positions. So, if we can occupy these three facial positions for these reactions, we can expect, we will see definitely for such reactions on vanadium model compounds. So, if we can have this, so if this is the position, this is the position and this is the position. So, one face, this is the face, trigonal face, which can be occupied, and once the trigonal face is occupied, we use one particular type of ligand, we call it as a facial ligand, like this very useful facial ligand is the triazacyclonone, TACN, we call it as a TACN ligand. Triazacyclonone ligand, when it reacts or giving some vanadium compound, so definitely when it reacts with say simple vanadium trichloride, what compound we do expect, because we will see in our some next classes that how these model compounds can be generated and can be a very good catalyst. So, we have 3 nitrogen donors from here, rather NH donors. These 3 are forming on a face 3 bonds on the vanadium, and we can have 3 chlorine bonds to the vanadium, giving rise to a beautiful vanadium compound bound to TACN ligand and the 3 chlorine units. So, this is the TACN part. So, if this is the thing, that means these 3 positions we require. So, these 3 are catalytically active sites. So, they are because all these peroxides and all other groups, the OH functions, they are mostly covering these 3 sites. So, they are the catalytically active sites and all the reactions are going from these 3 positions. That means, the other face of the octahedron. So, if we consider the face is forming from the octahedral geometry, so this basically going for this particular type of octahedral geometry. So, other face, this is one face and this is the other C3 face of the octahedron. So, other C3 face of the octahedron is basically giving that particular type of reactivity. So, in the final step therefore, for the catalysis, we just can go for the removal of HOCl. So, HOCl is removing, hypochlorous acid is removed, and the original catalyst in the native state is getting regenerated. So, if we see the entire catalytic cycle, the mechanism for the vanadium chloroperoxidase activity, we see that when we do not give any other organic substrate or the very complicated organic molecule from the marine origin, we do not get the corresponding chlorinated product of the organic compound. So, if the organic substrate is lacking, that means the organic substrate is not there, what we see that the water molecule is getting attacked by these particular reactions of this thing. That means, when hydrogen peroxide was there, the Cl is there. So, HOCl is forming over there and this HOCl is not getting any organic substrate to react to get the corresponding chlorinated organic product. Instead, this hypochlorous acid is leaving from the medium as HOCl from the reaction system, and the native catalyst is getting generated. So, this particular one, when we see that this cases, that means the vanadium site is there. So, the important step for these reactions is that vanadium is forming the corresponding peroxo linkage. So, which is very important to note. So, the formation of this vanadium peroxo linkage, that means this one, as well as sometimes this is forming the corresponding oxoform, that means whether this oxoform is there, that means this one is in the form of oxo. So, VO2 and the peroxo linkage. So, the NMR data what we can have, because these two oxygens what we have, these are not changed. So, dioxo-vanadium center when attached to the peroxo species. So, this particular entity, the entire this particular entity can be analyzed through the vanadium 51 NMR. So, if we can study the corresponding vanadium 51 NMR, it can immediately indicate that this vanadium center is bound to the peroxo linkage as well as to oxo as the vanadium dioxide. So, NMR data is definitely indicating us that we have the definite binding of the peroxide, and the vanadium dioxide unit is attached to the O2 species, but how it is reacting with the Cl minus. So, how Cl minus is reacting with the active vanadium center attached to the peroxide is not known from the NMR data. So, we do not get any direct evidence for binding of the Cl minus to the vanadium center, because at this particular point the Cl is not attacking to the vanadium center, and it is at the same time not giving the vanadium Cl bond. So, at the rate determining step or the rate limiting law, what we can have is found where the nucleophilic attack of the halide ion, that means this Cl minus is only functioning as a corresponding nucleophilic attack on the protonated protein peroxide complex generating X plus. So, at this particular point only the step which is very much dependent on the formation of the vanadium peroxo species as well as the attack of the halide anion, and we have the corresponding protein peroxide complex, that means the protein which has the vanadium center, and that vanadium center is responsible for the formation of the corresponding peroxide linkage, and that is then reacting with Cl minus and forming Cl plus. This Cl plus is basically reacting with the hydroxide anion forming the corresponding HOCl or the hypochlorous acid. So, during these oxidation processes, which can be also true if our X is Br or X is I. That means, for bromide oxidation or for iodide oxidation, the same peroxide we are utilizing. So, if we compare the corresponding redox potential for the Cl minus, Br minus and I minus, we find that the corresponding oxidation potentials of the halides are pH dependent, and in the catalytic cycle for all these reactions also we see that the pH dependence is also very much crucial, whether we are going for protonation of the vanadium OH bond or the corresponding thing what we are getting, that the deprotonation of the OH from the vanadium center to giving the vanadium oxo center. So, more acidic pH values are definitely required when we try to oxidize the more electronegative halide. So, if we can have the species, that means Cl minus, Br minus and I minus, which we all try to oxidize. So, we basically try to oxidize all three in presence of the H plus. So, initially we all know that if some suitable oxidizing agent is available, we get chlorine, we get bromine, we get iodine. Not only these, but also we want to have Cl as Cl plus, Br as Br plus and I as I plus. So, this formation of these things, so they are very much dependent on the different E0 values, and these E0 values are also dependent on the condition, that means the H plus availability, that means the pH of the medium. So, when we try to oxidize the Cl minus to get Cl2 or Cl plus, we should have a corresponding very acidic pH value for the more electronegative halide oxidation. So, the enzyme must have some suitable mechanism, it has some flexibility in this enzyme system that they basically modulate the different E0 values, because the reagent is fixed, we have the vanadium center, we have the peroxides, but the enzyme should have some flexibility such that depending upon the source of the enzyme, it definitely modulates the different E0 values. So, for the different enzymes we have the different E0 values for the oxidations of Cl2, Br2 or I2. Okay. Thank you.